welcome to my channel ANUPAMA Biology Classes. In this video, our topic is transpiration. This video is taken from Plant Physiology series, CBSE class 11th, part 7th of chapter 12. Now, let's see here the lecture overview. So in this video, we will focus on transpiration, types of transpiration, mechanism of water loss, stomatal transpiration, mechanism of stomatal movement, in which starch-sugar hypothesis, proton transport concept, and the last is blue light stimulated stomatal opening. Now, let's come on the first topic, which is transpiration. So, what is transpiration? The loss of water in the form of vapors from the living tissues of the aerial parts of the plant is termed as transpiration. If you see the overview of transpiration, water is passively transported into the roots and then into the xylem. The forces of cohesion and adhesion cause the water molecules to form a column in the xylem. Water moves from the xylem into the mesophyll cells, evaporates from their surface and leaves the plant by diffusion through the stomata. So practically, most of the water absorbed by plants is ultimately lost into the atmosphere. Only a very small fraction of water, generally less than 5%, is utilized in plant development and metabolic process. Basically, it is an evaporation phenomena, but it differs from the general process of evaporation. Evaporation is referred to the loss of water vapor from a free surface, whereas in case of transpiration, the water passes through the epidermis with its cuticle or through the stomata. If we talk about the magnitude of transpiration, as far as the magnitude of transpiration is concerned, Mayer in 1956 had reported that some of the herbaceous plants, under favorable conditions, transpire the entire volume of water which a plant has, and it is replaced within a single day. This means that of all the water absorbed by plants, approximately 95% is lost by transpiration and 5% or less is used in the plant. If it were not for the loss of water by transpiration, a single rain or irrigation would have been provided the enough water for the growth of an entire crop. Now, after this, types of transpiration. So how many types of transpiration? There are three main sources through which water loss from a given plant can be affected. So these are stomatal transpiration, cuticular transpiration, and the third is lenticular transpiration. So in case of transpiration, the water eventually is released to the atmosphere as vapor via the plant's stomata, which is tiny, closable pore-like structure on the surface of leaves. Overall, this uptake of water at the roots, transport of water through plant tissues and release of vapor by leaves, this all is known as transpiration. So on the basis of passage through which plant gives out water in the form of vapor, transpiration is of three types. First one is stomatal, second is cuticular, and the last is lenticular transpiration. So in case of first type, that is stomatal transpiration, it is the loss of water which occurs through specialized apertures which are present in leaves called stomata, which you can see in this picture. In herbaceous plants, the stomata can also occur in the epidermis of green stem. It accounts for 80 to 90% of the total water loss from the plants. Second is cuticular transpiration. The cuticle provides a relatively impermeable covering. If it is thin and green, up to 20% of the total transpiration may take place through it. But as its thickness is increased, the extent of water vapor loss is significantly reduced. And the last one, that is the lenticular transpiration. Water is lost through the lenticels of the fruits and woody stems, although to a much less extent than through stomata. Such a transpiration is termed lenticular transpiration. Now, here you can see what are the main differences between the stomatal transpiration, lenticular transpiration and cuticular transpiration. In case of stomatal transpiration is the evaporation of water through stomata. Lenticular transpiration is the evaporation of water through lenticels, and the cuticular transpiration is the evaporation of water through cuticles. If we talk about the plant surface, in case of stomatal transpiration is stomata, in case of lenticular transpiration is lenticels, and in case of cuticular transpiration is cuticles. In case of percentiles from total transpiration loss, in case of stomatal about 85 to 90% of water loss. In case of lenticular 0.1% of the total transpiration loss, and in case of cuticular transpiration 5 to 10% of the total transpiration loss. According to the occurrence, stomatal transpiration occurred during the daytime, lenticular transpiration occurred throughout the day and night, and the cuticular transpiration also occurred throughout the day and night. After this, mechanism of water loss. So, if we talk about the mechanism of water loss, the leaves absorb visible, that is light, and invisible, that is infrared radiations from their surroundings during the daytime. At night, they absorb heat from the surrounding air. Increase in the temperature due to the radiant energy during the daytime and due to heat energy during the night cause vaporization of water from the cells of transpiring plant organs, that is leaves. Vaporization or evaporation of water is a powerful cooling process. It reduces the temperature of plant organ about 2 to 5 degrees centigrade below that of the atmosphere. During vaporization, the target cells of parenchyma lose water into the intercellular space. As a result, the intercellular space gets saturated with water vapors. The atmospheric air, on the other hand, is dry. The waxy cuticle on the leaf surface restricts the loss of water vapor. However, the water vapor diffuses out from the intercellular spaces into the dry atmosphere mainly through the openings between the guard cells of stomata. A minor amount of water is lost directly from the open surface of leaf through cuticle. The loss of water vapors continues till the stomatal apertures are open. Therefore, transpiration is regulated by opening and closing of stomata. Now, after this, let's see the main mechanism of transpiration through stomata, which is known as stomatal transpiration. So, what is stomata? The stomata are teeny pores present in the epidermal surface of leaves, young stems, and in certain fruits like banana, citrus, cucumber, etc. They are also found in almost all the young aerial parts of the plant body. A typical stoma is microscopic and usually consists of two kidney-shaped guard cells surrounding a pore. The plural of stoma is stomata. The guard cells are usually much smaller in size as compared to other epidermal cells. They are therefore rapidly affected even by a small change in turgor. A stomatal pore is generally elliptical in surface view. The dimension of stomatal pore varies from species to species, but on an average, it measures about 20 micrometer long and about 10 to 20 micrometer wide when fully open. In some species, the two guard cells are surrounded by accessory or subsidiary cells which differ in shape and size from other epidermal cells. Guard cells generally have thick walls towards pores and thin walls on opposite side. Moreover, the cellulose micelles in the guard cell walls are oriented radially rather than lateral. The guard cells walls have special elastic properties. The adjoining cell walls of the two guard cells around pores are free and not attached with each other. These properties help them to stretch laterally during the stomatal opening. Now, if we talk about the physiology of stomata, the guard cells differ from other epidermal cells in mainly two respects. First one is they contain chloroplast and perform the phenomena of photosynthesis, and the second is they have a special type of wall thickenings. The photosynthesis in guard cells differ from normal photosynthesis of the mesophyll chloroplast. The guard cells are heterotrophic. However, starch is synthesized in the guard cell chloroplast by the sugar transported from the adjacent mesophyll cells. The guard cells are characterized by accumulation of starch during the night in dark and their degradation during the day in light. The mesophyll cells, on the other hand, accumulate starch during the day and decrease during the night. This property helps in the opening and closing of stomata. Now, here in this picture, you can see that the structure of open and closed stomata. That means in case of the stomata open, guard cells are swollen, and in case of stomata closed, guard cells are shrunken. Now, after this, see here the mechanism of stomatal opening. So, what is the mechanism of stomatal movement or opening? It means opening and closing of stomata. A variety of external and internal stimuli affect the opening and closing of stomata by altering the size of stomatal pores. The important among them are the light and dark, carbon dioxide concentration, water supply, pH of the cell sap, etc. In most of the cases, when water supply is adequate, the stomata tend to open during the daytime in response to light and close at night. The opening and closing of stomata operates as a result of turgor changes in the guard cells. When the guard cells become turgid, their thin walls get extended and the thick walls become slightly concave so that the stomatal aperture opens. On the other hand, the guard cells become flaccid when they lose water. Their thick walls revert back to original position, resulting in the closure of the stomatal pore. Investigations on submicroscopic anatomy of the guard cell wall suggest that the spatial orientation of the cellulose microfibril and micelles are mainly responsible for opening and closing of stomatal pores. These studies have shown that cellulose microfibrils and the micelles are arranged around the circumference of the elongated guard cells, and this arrangement is called radial myceliation. Here you can see that in the picture, that means radial myceliation. Such guard cells, when take up water and expand, cannot increase much in diameter because the microfibrils do not stretch much in elongation. On the other hand, they increase in length. Since the two guard cells remain attached to each other at both ends, they bend outward on swelling and result opening of stomatal pore. Now, there are two main theories about the mechanism of stomatal movement, in which the first one is starch-sugar hypothesis. So, this theory was formulated in 1923 by J.D. Savre and modified by Stewart in 1964. If you see here, an outline by this theory is photosynthesis in case of light occurs in light, so which consumes the respiratory carbon dioxide present in the intercellular spaces. This lowers the hydrogen ion concentration of cell sap and pH of guard cells is increased. So, because of this, high pH favors the activity of enzyme phosphorylase, which converts starch into glucose 1-phosphate. Here you can see here. Then the glucose 1-phosphate is further converted to glucose 6-phosphate by enzyme phosphoglucomutase. After this, the glucose 6-phosphate is converted to glucose and phosphate by enzyme phosphatase. The glucose and phosphate units get dissolved into the medium and increase the concentration of cell sap. This causes further increase in the osmotic pressure of the guard cell and consequently its water potential is decreased. This results in the movement of water into the guard cells from the surrounding cells. The guard cells become turgid and swell, thus the stomatal pore becomes open. Now, in case of dark, the photosynthesis does not occur in dark. The level of respiratory carbon dioxide in some stomatal cavity is increased. This result decrease in the pH of guard cells. At low pH, the glucose molecules are converted to glucose 1-phosphate, making use of respiratory ATP in presence of enzyme hexokinase. The glucose 1-phosphate molecules are converted to starch in presence of enzyme phosphorylase. So, the synthesis of starch leads to the dilution of cell sap by consuming its dissolved glucose molecule. Thus, the osmotic pressure of the cell sap is decreased and its water potential is increased. The guard cells then lose water to the surrounding cells and become flaccid. The stomatal pore is then closed. So, in case of light, carbon dioxide concentration decreases. Because of this, malic acid formed in guard cells so dissociates into malate ions and hydrogen ion. Then potassium ion are taken into guard cells and hydrogen ion are transported out of the guard cells. Because of this, osmotic pressure of the guard cells increased, then endosmosis into guard cells become turgid and stomata open. While in case of the dark, carbon dioxide concentration increases due to decreased pH of guard cells, ABA released to stop the potassium ion exchange. Malate ions present in guard cell cytoplasm combine with hydrogen ion to form malic acid. Then potassium ion transported back into subsidiary cells, resulting in decreased osmotic concentration of the guard cells. Because of this, exosmosis from guard cells become flaccid and stomata close. So this is all about the opening and closing of stomata with the help of starch-sugar hypothesis. So, starch-sugar interconversion theory, the effect of pH on starch phosphorylase enzyme during day and night. Starch phosphorylase reversibly catalyze the conversion of starch plus inorganic phosphate into glucose 1-phosphate. During day, high pH, starch converted into glucose 1-phosphate, then lower osmotic potential, then target guard cells, the stomata open. While in case of during dark, low pH, glucose 1-phosphate converted into starch, high osmotic potential. Because of this, flaccid guard cells and the stomata close. So this is all about the starch-sugar interconversion theory. Now, see here the criticism of the starch-sugar hypothesis. So what are the criticisms? A starch-sugar interconversion is a slow process which cannot account for rapid stomatal movement. Starch or other polymerized polysaccharide do not occur in onion plant where stomatal movement occurs. Glucose is not detectable in the guard cells when stomatal opening occurs. The theory could not explain the extra effectiveness of the blue light at the time of stomatal opening. So this is all about the criticism of the starch-sugar hypothesis. After this, the second concept is proton transport concept. This theory was proposed by Leavitt in 1974 and elaborated by Reschke and Bolling. So here you can see a brief account of this theory. In case of light, the starch content of the guard cells disappear in light. First of all, the starch is converted into organic acids, particularly phosphoenol pyruvic acid. Phosphoenol pyruvic acid then combines with the carbon dioxide to produce oxaloacetic acid and then to malic acid. The organic acid like malic acid dissociate into malate anion and hydrogen ion in the guard cells. Hydrogen ion are transported to the epidermal cells, particularly the subsidiary cells, and potassium ion are taken into the guard cells in exchange of hydrogen ion. The process is called ion exchange. Potassium ions are balanced by the organic anions. Some chlorine ions are also taken in to neutralize a small percentage of potassium ion. So hydrogen ion potassium ion exchange is an active process which requires involvement of energy supplied either by the respiration or photophosphorylation. Increased concentration of potassium ion and malate ion in the vacuole of guard cells causes sufficient osmotic pressure to absorb water from the surrounding cells. So, increased turgor of the guard cells due to entry of water results stomatal pore to open. Now, in case of dark, carbon dioxide concentration is increased in dark in the substomatal cavity because photosynthesis is stopped but respiration is continued. Higher concentration of carbon dioxide in substomatal cavity prevents proton gradient across protoplasmic membrane in guard cells. As a result, active transport of potassium ion into the guard cells ceases. So, according to Cowan et al., which is proposed that the closure mechanism involve participation of an inhibitor hormone abscisic acid which functions at lower pH. As soon as the pH of the guard cell decreases, the abscisic acid inhibits potassium ion uptake by changing the diffusion and permeability of the guard cells. Malate ions present in the guard cell cytoplasm combine with the hydrogen ion to produce malic acid. Excess of malic acid inhibits its own synthesis by decreasing the activity of PEP carboxylase. This changes causes reversal of ion movement so that potassium ion is transported out of guard cells into the surrounding epidermal cells. The osmotic pressure of the guard cell is thus decreased. This results in the movement of water from the guard cells to the surrounding cells. The guard cells become flaccid and the stomatal pores gets closed. In fact, the actual mechanism of stomatal closure is still not clearly known. So, in case of control of stomatal opening and closing, guard cells take up potassium ion by active transport. This causes water to enter the cell by osmosis. Here you can see the stomata opening and stoma closing. Now, the third theory, that is the blue light stimulated stomatal opening. Studies of guard cell physiology reveals that red light induces accumulation of sucrose that causes osmoregulation, and blue light plays an important role in stomatal movement by changing the osmoregulation of the guard cells. Blue light is absorbed by a carotenoid zeaxanthin which initiates the process of stomatal movement. So this is the end of video in which we know about the transpiration, types of transpiration, mechanism of water loss, stomatal transpiration and mechanism of stomatal movement. Now the next upcoming video will be on guttation. So if you understand this video, please like and share it and subscribe to my channel ANUPAMA Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.